The overall goal of this procedure is to elucidate the protein expression profiles of liquid and solid tumors. This is accomplished by first enriching cancer cells from patient-derived blood or tissue samples. In the second step, proteins are isolated from the cancer cells and then mixed with a tumor-specific Super-SILAC quantification standard. In the final step, the protein mixture is digested. Ultimately, mass spectrometry is used to show the protein expression profiles from liquid and solid tumors.
This method can help to answer key questions in the oncology and pathology field, such as how can drug targets and mechanisms of therapy resistance be identified.
Though this method can provide insight into biomarker discovery, it can also be applied to other systems, such as the investigation of diverse solid and liquid cancers.
Demonstrating the procedure will be Dr. Hanibal Bohnenberger, Institute for Pathology, and Dr. Christof Lenz from my laboratory.
To prepare liquid tumor cells for fluorescence sorting by flow cytometry, begin by aliquoting 1 milliliter of the human mononuclear cell sample suspended in PBS plus 2% FBS for the negative control and 1 milliliter for each fluorescent dye used in the final stain. Next, incubate the cells with the appropriate antibodies for isolating leukemic cell population and for the compensation controls on ice. After 30 minutes, wash the cells twice with PBS plus 2% FCS. Then add a cell viability stain to the appropriate tubes and sort the cells according to the schematic, collecting the